Hello, General Biology, and welcome to the third in the screencast series on photosynthesis, Chapter 8. And we're going to watch this 7-minute video, which kind of gives an overview of both the light-dependent and the light-independent reactions, and also, again, explains that electromagnetic spectrum a little bit. And then we will get into the light-dependent reactions after this is over with. So please sit back and watch.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
All right, what did you think of that video? So it's a little complicated, and you're probably thinking, oh, I'm never going to get this, but you will, and I'll help you through it. So we're going to just focus on the light-dependent reactions first, and the light-dependent reactions are going to happen within the thylakoid membrane, and that's inside of the chloroplast. And it's going to use something in the membrane called the electron transport chain. So here's our chloroplast, and we're going to take one of those little discs out that we call thylakoids, and we're going to look at it. And here it is. It is a phospholipid bilayer, which you already are familiar with. So you can see the phospholipids here, and it's a bilayer, so there's 2 layers. The purple blobs that you see in here are proteins. This one happens to be a special kind of transport protein that we're going to talk about. It's called ATP synthase, and synthase means to make, so it's going to be making ATP. These other purple blobs that you see are transport proteins, and they're going to, the smaller ones are just going to transport electrons, but this larger one is an ion pump, and it's going to pump ions, specifically the hydrogen ions that you see here. And it's a pump, which means it's going to work by active transport, because notice, inside of this thylakoid, this inner thylakoid space, which is called the lumen, there's a high concentration of hydrogen ions. But on the outside, there's a low concentration. So the pump's job is going to be to move the hydrogen ions from a low concentration to a high concentration. And that's going to require energy. they're going to get that energy from the electrons. Remember I mentioned that water molecules split, and when they split you get hydrogen ions and you get electrons. Well, electrons are going to be that battery pack of the ion pump, and when they get to that ion pump they're going to power the ion pump to move the hydrogen ions into the lumen against the gradient. Okay, so that's what those purple things are. Now notice that there's two green blobs, and these green blobs are called photosystems, and their purpose is to trap the energy of sunlight and use that energy of sunlight to either split water or to re-energize the electrons, recharge them, so that they can power the pumps and move on to make ATP. So we have two photosystems, and this one is photosystem 2 and this one is photosystem 1. And they're named that way by the way that they were discovered. But the process starts with photosystem 2. And that is where the light energy is going to be coming in. It's going to be trapped in the tiny little pigments of this photosystem called chlorophyll. And that energy trapped in the chlorophyll is going to be used to split the water. When we split water, we're going to get hydrogen ions, we're going to get electrons, and we're going to get oxygen. The oxygen we're not going to worry about because it's going to leave the cycle. But the hydrogen ions, some are going to collect in the lumen, where others are going to collect in the outside. The electrons, they're going to remain for just a moment in photosystem 2, and as light energy continues to be absorbed, it's going to energize those electrons. And those electrons will then move to the first transport protein, and then into the second transport protein, which we already described as an ion pump, and it'll turn on the pump. They're kind of like the battery pack for that pump, and now they'll turn on that pump to move the hydrogen ions against the gradient. When they're done, they'll move into another transport protein and then into photosystem 1, where they will be re-energized, and then they'll go to another transport protein where they will be picked up by the electron carrier, NADP+, and they'll pick up some hydrogens and the electrons and become NADPH, and will go off to the electron transport chain. Then we have the second part of the electron light-dependent reactions, and there's this ATP synthase. And we notice that there's a high concentration of hydrogen ions. ATP synthase works by facilitated diffusion. So that means that by natural forces, the hydrogen ions will move from an area of high concentration to an area of low concentration through this channel, this transport protein. When they do, it'll cause this ATP synthase to spin. It's going to spin around just kind of like a spoon stirs things. It's going to spin around, and if there's ADP and independent phosphate groups located in the stroma, it'll pull these two together with that stirring motion and make ATP. So we wind up getting NADPH and ATP. So in our next screencast, we're going to look at this. I know you're saying, oh, Mrs. Saunders, I don't get it, but we're going to look at this again in more detail. So we're going to end it here, and we'll come back to that electron transport chain.